Okay, so proteins and our genetic code. So, how the genetic code creates proteins is what we're going to be talking about in this sequence. So, we talked um, about transcription. So, this is in mRNA, how we get from DNA to RNAs or mRNA, um, whichever one, actually, all RNA. Is through transcription. It's like writing a prescription for what you need to happen, right? So the DNA transcribes into RNA. So one gene or one segment of that DNA is transcribed into mRNA, and then the mRNA goes to the ribosome. And the next process is called translation from RNA to protein. We'll talk more about what translation is in a moment. However, there's a code to this. So going from RNA, which is a nucleic acid, to protein, which is an amino acid, these are very different structurally, and we have to have a way to convert or translate between the two. And the way that we do this is through codons. So codons are sequences of 3 nucleotides which specify an amino acid. So for example, having those three nucleotides in a row codes to say you need a phenylalanine. Codons in mRNA are recognized by anticodons in tRNA. So we talked about anticodons in tRNA just a moment ago up here. So these are opposite to the codons that we're talking about now. So codons are in mRNA, anticodons are in tRNA. So they're opposite and they recognize one another. Okay, so there's a chart for this. There are 64 codons, and once you put together all of the different combinations of how you can get, or different 3-letter combinations of those 4 letters, and that is how you incorporate the 20 natural amino acids into proteins. So for example, we said UUU here is codes for phenylalanine. Or let's go to the something random in the middle. ACC codes for threonine. So this chart just tells you, that's how you read it. You look for the different ones. The first letter here is going to be on this side. So all of the ones that start with U are in this top portion, all the way down. Your second letter, UCAG. So looking at the second letter, so this whole column is a U column, this whole column is a C column in the very center. And then lastly, the third letter is on this third side. So you get all of the different combinations there. And it codes for all of the different amino acids. There are special things though. There are 3 stop codons. So here, UAA, UAG, and UGA tell the replication to stop, tells the translation to stop. And then additionally, AUG tells the translation to start, or it also translates for methionine. So let's do an example of this, how we're actually going to use this. So what is the sequence of amino acids specified by the following codons in mRNA? We'll get to the second part in a moment, but if I'm given this code of mRNA, how I need to figure out the protein sequence. So what I would do is I would go and I'd say GUC and go up into this chart and I'd find G and then I'd find U. So I know I'm in this area here, GUC. That tells me I need a valine. So my first amino acid is a valine. AGC is next. A here, G here, C here. So arginine. Oh, no, AGC, serine. There we go. I saw the G and thought it was a C. AGC is serine. Then we have, you just keep going through. And in this case, you find out that the last one is a stop. So this is the overall protein sequence that you're given based off of this mRNA strand that you're told. The second part says, what is the sequence of DNA that this mRNA strand was transcribed from? So what we're going to do is very similar to what we did when we were replicating DNA, in that you have the opposite of those complementary base pairs in the opposite order. So we have 3' here, or 5' in RNA. So you'll have the 3' going backwards. And then you're going to use your complementary base pairs. So if this one has a G, this one has a C. This one has a U, so now it has an A, because remember in RNA, it's a U. We keep going, we have C to G. Now we have A here, and the tricky part is that we need to remember that in DNA, we have a T, not a U. So the base pair for A would then be T, not U. So being careful to make sure that you're using T or U correctly is important. So we'll just go through, you go through all of those base pairs and write it out that way. Okay, I think that's where this video is going to stop.